All right, so we've gotten through a lot of material in this chapter where we've been able to take a look at this process of DNA being used as it's replicated so that it can get passed on to the next generation of cells. This is what's known as the vertical transmission, and we can also look at the DNA in terms of what it actually does or what it's used for, because it does code for those functional products. So if we look at this process when we're considering a cell that's out there, we can see this horizontal gene transfer where the DNA is moving from the parent cell to the daughter cells, but we can also see this process in terms of the vertical gene transfer where it's going from a donor cell to a recipient cell that is out there. And so this is a slightly different process that we're looking at here, but it is something that is going to be very important because we can find examples of where it occurs in prokaryotic cells. And so we can look at these three different things that are out there, this process of vertical gene transfer in transformation, transduction, and also conjugation. And it's important to remember that when we're looking at this process, the more closely related the cells are, the more successful or the more highly found this process is going to be. So if you're looking at gram-positive cells, more likely to occur between relatives there versus between gram-positive and gram-negative cells, it's less likely to occur because there is enough of a difference at the genetic level that it's not quite as successful. And effectively, your cells need to have the donor and the recipient, and we'll see that the donor doesn't always have to be alive, but also you have to have recombination occur between these cells for that DNA to actually be successful there. If you take a look at the figure from the book, we can see this gene transfer process sort of highlighted in these three little cartoons here. We've seen this process of transformation from some of the lab stuff that you worked on this semester. And here you've got a naked piece of DNA, so that could be from a dead donor cell that's out there that is decomposing, and there's a chunk of DNA just floating there. And these cells have this ability to take up this DNA directly from the environment there. And then that DNA could get incorporated or recombined into the chromosome, and now when that cell divides, the chromosome will pass on those genetic characteristics that the cells have picked up. So that idea of Griffith's experiment that he first found the transformation of Streptococcus pneumoniae out there because they were picking up DNA from the dead donor cells, the heat-killed smooth cells. In lab, our example used a circular piece of extra chromosomal DNA known as a plasmid, but this does indeed transform the cells because they have different characteristics after the process. The second one that we can see here is this process of transduction. And this one is different because transduction, your DNA is actually going to be carried inside of a virus from the donor to the recipient cell. And in this video, we'll be able to take a look at this process and what's going on, sort of an overview here. So if we click on that little guy, you can see there is your bacteria that infected this donor cell right there. And that donor cell, the bacteriophage actually resulted in the production of enzymes whose job is to basically chop down the host chromosome DNA. And if you remember from the lab, when we look at the restriction enzymes, those restricted the growth of the bacteriophage because they chopped up the viral DNA that was injected into the cells. So you are literally looking at a process where these two cells are shooting enzymes at each other, and whoever hits the DNA of the other one first is the one who wins the battle there. Well, here in this case, we can take a look at this DNA chunk that came out of there. In this example, it happened to get packaged inside of that virion. And so this could just be a random piece of the chromosome, and then the virus proteins that were made inside this donor cell, they just happen to package that chunk of DNA right there. And so now you've got a virion that is carrying some DNA around, and it goes out to our recipient cell right here. And so now this virus has, really it's just a shell. It's not carrying the infectious viral material there, but it is shuttling that piece of DNA from the dead donor cell into this live cell right here. And so this is actually a fortuitous infection of the virus, because now this recipient cell can pick up that DNA. And if you remember, this is sort of generalized transduction. You can also have specialized transduction, where you have a prophage when it excises from the chromosome because the cell is stressed out. Then that virus carries a chunk of the host chromosome with the viral DNA. And so it's a little bit different process. Here you're basically taking a big chunk of the chromosome that's left over floating around in this cytoplasmic bag as the virus replicates there. So kind of an interesting process to look at. So in both of these cases, the donor cell is dead, or the husk of the donor cell there. But in our third case with conjugation, you actually have a live donor cell and a live recipient cell. And to do this process, gram-positive cells basically will stick together using that sticky glycocalyx or capsule layer on the outside. And then you can actually have the physical transfer of the DNA between these cells. But with gram-negative cells, this is where the pilus comes into play. And if you remember, that is a long strand of pilin protein that is on the outside of the cells. And here the bacteria actually uses that pilus to lasso another cell and then bring it over so that it is close by the donor cell. And if we take a look at this process here, we can see it showing in this little video right there. There is your pilus that has brought over this cell, and our donor cell right here has these little appendages on the outside, but it also has a plasmid right here, and this plasmid has the pilin gene. So this cell is known as an F+ cell. It has what's known as the fertility factor, and it is mating with this cell over here that is lacking the plasmid. So it is lacking the fertility factor, or it's an F- cell right there. And during this process, what happens is it actually makes a copy of one strand of this DNA, and that strand is sent over from the donor to the recipient cell. Now this cell is transformed as a result of this conjugal visit, which is really what's going on, because these cells have a donor and a recipient. This is a form of bacteria sex, so that the cells that come out of this process are changed genetically. But notice, as a result of this, both of these cells are F+. So you can almost think of it as this one has changed from the female version, which is the recipient, to now this is the donor version. So you start out with a male and a female, and you end up with two male versions of this, complete with the beard on the two cells right there. So in this process, these cells have been changed as a result of conjugation there. And that cell can continue to donate that DNA to other cells that are out there. So imagine this is E. coli in your intestines. You've picked up an antibiotic resistance plasmid. One cell has it, and then it is able to basically conjugate with all these other gram-negative bacteria in your intestines. And now you've got this population of antibiotic resistant bacteria that have picked up not only the plasmid with the pilin gene, but what if you actually have that antibiotic resistance gene on your plasmid there as a result of this process. You could also imagine something where you get transformation that gets the DNA into the cells, and then it recombines with the plasmid, or even with the chromosome. And sometimes there is this origin of transfer built into the plasmid where they start it. If this plasmid gets recombined with your chromosome, you can actually get the chromosome being transferred through conjugation from one cell to another cell. And by doing experiments with F+ and F- cells that are out there, they were able to actually determine it takes about 100 minutes for these bacteria cells to transfer their whole chromosome from one cell to another one. There is another example that doesn't really involve the donor or recipient, but it can result in changes in the DNA, and this is what's known as a transposon. And they're kind of an interesting little structure here. If you look at a transposon, what you will see is they actually have a sequence of DNA here that has what are known as inverted repeats on either side there. And then in the middle of what's known as the cassette, you have a gene that codes for an enzyme called the transposase. And so you can look at this little thing right here. You've got a typical transposon that has its inverted repeats in the DNA right there. And the transposase enzyme is produced, and it actually binds to these inverted repeats right here, and that causes the DNA to loop out right there, and then the transposase enzyme can chop out your DNA from there, and that DNA will match up with random locations in another piece of DNA out there, and it'll insert your cassette in between these inverted repeats into that DNA. And in this example right here, what we see is that gene B over here has been disrupted, where gene A that was over here where it was originally, maybe it got restored to its natural site, maybe the transposase screwed it up and it's got some mutations, but in this one you've definitely inserted a chunk of DNA into the middle of that DNA, or into the middle of the gene in the DNA, and that could result in a mutation. And if we look at this process here, it's kind of interesting to see. Here is a little example showing you there's your inverted repeats with your transposase, and this little cassette right here is just jumping randomly around within this piece of DNA. So imagine you've got a chromosome there, and the transposase inserts that piece of DNA into a plasmid, and then this cell does conjugation and it transfers that DNA to another cell. Or maybe you've got transformation that occurred that moved that DNA from one cell to the other. Well, now you've effectively shuttled your piece of DNA from one cell to another cell out there. And if we look at this example, this is a more complex version where now you've got two cassettes on either side of an antibiotic resistance gene. The transposase recognizes the two outer repeats right there, and so it cuts out this whole chunk of DNA. Well, now you can shuttle that piece of DNA from your chromosome to your plasmid. So rather than the transposon being moved, now it's the transposon and these other genes. And so this could result in the changes in the genes of a cell that is out there. So slightly different mechanism from what we saw with the other methods, but this is something that is definitely happening out there.